Julianne, will you just give me a heads up when we're live?
We are live now.
Good evening, everyone.
Hi, everyone.
Welcome to another. Hi, everyone. If you can just please give a thumbs up in the comments section that you can hear us. And then I'd just like to welcome you to another NERV. NERV is the Neural Engineering Research Venture. And myself, Siobhan, and Julianne will be your hosts for the evening. And you can keep an eye out for Darby in the comments. NERV aims to bring neuroscience to life in Africa and around the world. And we are really excited to have you join us tonight. Before we get going, I'd just like to run through some house rules. Just to make sure that it's a fair and pleasant experience for everyone online tonight. Please be mindful that we have a diverse range of views and always use respectful and inclusive language. Throughout the talk, please feel free to post comments. But if you have any questions, there is a specific Ask a Question feature where you can post your questions and then you can also vote for your favorite questions. Once the presentation is done, we will invite you on stage to ask your questions live. If you don't have a mic, don't worry about it. We will ask on your behalf. You can just add "please ask" to your question and then we will ask it for you. If your connection is slow, the best thing to do is to refresh the browser. And if your connection completely fails, don't stress. The talk is available to stream after the event. You can just follow the same link that you followed to join us tonight. And with that, I'm going to hand over to Julianne, who will introduce our speaker for this evening.
Hi, everyone, and welcome. I see in the comments there was mention of there being a bit of an echo with the audio. Can I maybe just get some indication if my audio sounds similar? Okay, some people are saying no echo for them. Okay. Welcome, like Siobhan said, to a new NERV. We are honored this evening to welcome Professor Jorge Mejias to the screen. Prof. Mejias is a principal investigator and assistant professor of computational neuroscience at the University of Amsterdam in the Netherlands. With a background in physics and mathematics, he obtained a PhD in computational neuroscience from the University of Granada in 2009 under the supervision of Prof. Joaquin Torres. Prof. Mejias has worked as a postdoctoral researcher in the labs of Profs. Andre Longton at the University of Ottawa and Ching-Ying Wang at New York University before joining the University of Amsterdam in 2017. His research interests include large-scale models of brain dynamics and cognition, short-term synaptic plasticity, dynamics of spiky neural networks, and multisensory integration. Without further ado, I am now very honored to welcome Prof. Mejias as our speaker this evening. Welcome, Prof. Mejias.
Thank you very much. First of all, I'm going to share my screen. Okay. So I guess you all can see my screen right now. Can you give a thumbs up if you do? Okay, I'm seeing thumbs up. So then I'm going to start with this. First of all, I'm going to start by thanking the organizers, Julian and Simon, for giving me the opportunity to give this very nice presentation. It's the first time that I present something online, so it's a new format. It's exciting for me. I'm nervous as a first-year PhD student right now, but I'm very happy to be able to share some of the work that I've been doing with so many people. So the work that I'm going to present is part of the work that I did during my last postdoc at New York University, and it's the basis of the work that I'm doing my new lab these days. But I'm going to focus on the work that is already published or released on bioRxiv and these things. And the idea, the main topic of the talk today is going to be computational models of large-scale brain networks, and I'm going to have I'm going to put a little bit of a focus on dynamics and a little bit on function, cognitive functions. So we can start by saying, for those of you who are a little bit familiar with computational neuroscience, which is nothing less than applying mathematical and computational models to study the brain and study how do we think. This is a field that has traditionally been focused on modeling and describing small systems. And by small, I mean neural networks that are typically a little bit of a part of our brain. Of course, one wants to start doing this, but eventually you want to move to try to model the entire brain or large chunks of the brain. And this has been comparatively a little bit more difficult traditionally for many reasons. Maybe one of them, maybe the main one, is that there was not a lot of information about how circuits are connected together at a large scale. So how different areas are connected with other areas in a very fine and very detailed way. While on the other hand, we had a lot of information about connectivity in small circuits. So this is kind of a problem. If you want to build a model of the brain that is realistic, you want to know exactly which areas are strongly connected with which other areas, or which connections are weaker than the others. And it has been traditionally very difficult to obtain this kind of information. So that's why the field has gone for a lot of time to try to model neural networks that are considered to be like small pieces, like a homogeneous neural network. For instance, I'm showing here two examples. A classical example of a spiking neural network, in which we have a network of different nodes, and each one of the nodes is a spike, is a model of a neuron. And you can see here with the red traces a neuron that is spiking, and then there's another one. So we have a full description of a neural network with spiking elements. We can, of course, try to simplify this model and go to what people call firing rate models, which are models that are more or less an average, give you an average of the overall activity of the system, so that you don't observe individual spikes of individual neurons, you observe the average firing rate of the whole population. And this has been traditionally useful to try to go beyond the models of local neural circuits. But still, even though we have the mathematical tools to do it, we lacked the anatomical data that we need to use to do this properly, instead of just putting this into a network without any kind of real information. And this has been easier and easier in the latest years due to the appearance of some structural data of large-scale networks. For instance, we can think about the marvelous work that the people at the Allen Institute are doing by building by basically building morphological data sets of cells, of individual brain cells for humans, for mice. We have also a lot of effort from the Human Brain Project that are trying to assemble large-scale models based on a lot of anatomical data as well. So slowly we are getting to a point in which we can go from single circuits, like the one that I'm plotting on the left, to large-scale circuits, like the one that I'm plotting on the right. One of the data sets that allows us to do this, and I'm going to be using this data set for the whole presentation, is the connectivity data from the cortex of the monkey, of the macaque. This comes from the Kennedy Lab, and they have been doing this study, recollecting and measuring the anatomical projections, the synapses from neurons in one area to neurons in another area. And they have a very rich data set that we can now use as a skeleton for our computational models. Okay, the data set has certain particularities. For example, this allows us to see whether the neurons going from one area to another area, projecting from one area to another area, are mostly in superficial cortical layers or in deep cortical layers. As you might remember, the cortex is divided, each area of the cortex is divided in different layers on top of each other, and then neurons can be breaking from one layer or another layer. And this reveals some kind of anatomical organization that I will detail a little bit later. But for now, since we have this anatomical data set, we are, of course, as computational modelers, we are, of course, very eager to start playing with this and trying to put computational models on top to see if we can find things about large-scale computations in the brain. With this, we are going to start with something very simple. We are going to start focusing on signal propagation in these large-scale networks and the importance of what we are going to refer to as the excitation-inhibition balance. And the reason I start with this is very simple. Before we understand how different areas communicate with or compute things with the help of each other, we need to find out how the information is transmitted from one area to another area. And this has been a classical problem in computational neuroscience that a lot of people have been working on for many years, and I'm putting some of the old classical references here. Most of these models would work in the sense that they have different, like a population of neurons here, A and then B and then C. We can identify each one of these neurons as basically neurons in one particular area. So then this basically describes a feed-forward model in which these neurons project to the neurons in the same area, in the next area, and these neurons project to the neurons in the next area, and so on. And they have done very interesting computational and theoretical studies about how an original stimulus produces some kind of a number of spikes, a volley of spikes, if you want. And these spikes can propagate across the different areas, or they can dissipate, like here on the right. The problem with all these approaches is that because of this lack of knowledge of the anatomical connectivity that I was mentioning at the beginning, these models are highly unrealistic in the sense that they don't reflect the exact connectivity of the brain. And this is a problem because we know that even though we don't know a lot about the connectivity of the brain yet, we know that that there's a lot of recurrent connections. So you have area A that is projecting not only to one area in a feed-forward manner, but it's projecting to many other areas, and these other many areas are projecting back to this original area. So there's a lot of recurrent connections. It's not a chain, it's something like a proper recurrent network, right? So what happens if we try to do this in a recurrent neural network? Well, we can find out right now. We can put these very simple models of activity propagation in the anatomical connectivity from the Kennedy lab that I just introduced a second ago. That I'm plotting here on the left in yellow. So this is the graph of our network, and in each one of these populations, in each one of the nodes, we have a population of excitatory neurons and a population of inhibitory neurons. And then we introduce an input in primary visual cortex, and we see what happens in this far away area over there. We see that things complicate a little bit, because if you can see here in blue, this is the response of the first area, V1. By the time this signal propagates and reaches the area 24C, this activity has dissipated a lot. And we can see how the response of 24C, if you see the y-axis is in logarithmic scale. So this is basically almost 4 orders of magnitude of difference. So this is pretty much means that nothing is arriving at nothing is arriving at the end of the hierarchy. You could tell me that, well, maybe the overall connections are very weak, and we can increase the strength of these interareal connections, and we can do that. But if we do that, we observe that the whole network enters into a positive feedback loop that basically feedbacks itself, and that means that we're going to have activity in B1 that is going to reach properly the activity in 24C, but then this activity is going to go back to the other areas, and at the end, all the activity in the brain is going to explode and it's going to grow until the maximum possible. And we know that this is not realistic, because this would, at least not in healthy terms, not in terms of a healthy brain, because this would constitute to what to an epileptic seizure, for example. So there's definitely something, and we are just moving the global coupling a little bit. So there's definitely a problem here. It means that in real recurrent networks, propagation of activity is either too weak to be useful or too strong that induces some kind of seizure state. And we know that this is because of the recurrent connections, because of the feedback connections, because if we take them out, we have a way smoother range of firing rates. So that means that the classical models of signal propagation in the feed-forward chains that I explained before are not useful when we consider a realistic network of the brain. Okay, so how do we solve that? What can we do to explain a reliable activity propagation that we observe in healthy brains, if we know that these feedback connections are so dangerous, are so disruptive for the normal functioning? Well, the solution that we found is to extend what is known the mechanism of transient amplification of weak signals that has been previously found in local balanced neural networks. And without going into much detail, I can tell you in a few sentences what this kind of balanced network and this transient amplification in balanced networks means. The idea is, if you see here, we have a classical local network of excitatory inhibitory cells. Just imagine that we have an input to the excitatory cell, and we have this response in green, and we consider that this response is very weak. Okay, so one of the tricks to improve the strength of this response without making the whole system explode and burst into never-ending activity is to reinforce the recurrent connections between excitatory cells, and at the same time reinforcing the projections from the inhibitory cells to the excitatory cells. And this would produce first a very strong transient response, and then once the inhibitory neurons kick in, they are going to shut down the activity. And then, as you can see here in the purple curve, it's going to be a response that is much stronger than the original one. And this is called a transient amplification that you can get in this kind of excitation inhibition balanced networks. Okay, so our idea was to try something similar, but instead of going for local systems, let's see if we can do something similar for networks that are connected via long-range projections. So imagine that we have here on the right area 1 here and area 2. The one possible way to do this equivalent scenario would be to reinforce the projections between pyramidal neurons or excitatory neurons in different areas, but at the same time reinforcing as well the local inhibition. Okay? So if we do that, we are going to obtain something very similar to what we have here. And this, in green, we have the original response of the model, and you can see how originally is a firing rate in this area of 100 spikes per second, and then by the time it reaches the last area, it's four orders of magnitude below. On the other hand, if we increase this kind of balance amplification, this kind of the balance between the excitatory and inhibitory cells, we see that compared to the green curves that I'm plotting here as well, it's just they look a little bit more flat because I'm changing the scale. We see that the new ones, the purple ones, behave way better. And we can see here how, compared to this case in which, and this is the worst case scenario, in which we basically have a fraction of activity, now we have like 1-2 spikes per second in the very worst case scenario. Many other areas we have 12 spikes per second, 5, 37. So it seems like the activity of the individual areas it's higher. So the signal is propagating better, and we can see here the maximum firing rate for each one of the areas that we are considering in our network. And in green we have the original result, and in purple we have the improved result once we use this transient amplification for balanced networks. And we see how this improves almost 2 orders of magnitude our previous result. So this is a very important mechanism to understand how neurons in different parts of the brain can communicate with each other without putting the whole stability of the network in a rough spot, right? So this efficient communication between different areas of the brain. And we can see how we go from this blue scenario in which the response is either very small, too small, or too large, to a situation like the black curve in which we have a smooth curve. We can, of course, try this with more realistic models, because right now the models that I'm showing you, they are the firing rate models that I was explaining at the beginning. They are averages of what's happening in one particular area. But you might want to check out what goes on when we consider a neural network, a model that has spiking neural networks. So we did that just to check that this finding that we have is robust and can happen in more realistic scenarios. So for each one of the areas, we are now including a neural network composed of 2,000 neurons per area, 2,000 spiking neurons like the one that I'm showing here. So this adds up to a total, since we have 30 areas, this adds up to a total of 60,000 neurons in our simulation. So we can now try if this works. And we can compute how the propagation of activity is in the control case, in the green case. And we see that there's these little dots are the spikes of neurons in each one of the areas. We see that there's a little bit of propagation, but then aside from this one and this one, it doesn't seem like the other areas care too much about propagating the signal. But when we increase a little bit the level of balance in the network, if we follow this strategy of transient amplification, we see that there's a lot more areas that are responding to this signal. You can see here in this simulated brain of the macaque, how in the case of a weak balance amplification, we only have activity here in visual areas. But if we reinforce this balance parameter, we have activity in other areas, including areas in prefrontal cortex and other areas. And we can see an improvement in many of the areas in our model. One particular thing before I go on that I want to mention is that we can also play with the strength of the input that we are giving to our system, and then we can see whether we need a stronger input or a weaker input to activate areas in different cortical lobes. For example, occipital lobes on the back of our head or prefrontal lobe on the front of our head. And we found that there's a significantly higher activation threshold for prefrontal areas compared to all the other areas in other lobes, meaning that you actually need a very strong signal in order to activate the areas in prefrontal cortex compared to other areas. And this has some connections with what people understand as the global ignition phenomenon that is one of the theories to explain conscious perception and consciousness. And I just want to mention that this little prediction of the model was found experimentally almost at the same time in an experimental study studying macaques and studying how macaques perceive visual inputs. And they also found that you need a very strong stimulus to have areas in prefrontal cortex, like dorsolateral prefrontal cortex here, respond to the original visual input that you are giving to the monkey. So this goes in the same line. It seems like the model gave a a good prediction that was later found experimentally, and it's kind of exciting because it's relating with a very difficult topic to study, that is consciousness. Now I want to follow up with the next logical step that we would take here, which is introducing a little bit more of anatomical detail into our model. In particular, we are interested in layers, and I will let you know a little bit more details in this sense in a second. As I was mentioning at the beginning, if we look at the anatomical structure of networks in the monkeys or in humans, we observe that there's some kind of order, some kind of hierarchy, in which we have early sensory areas like primary visual cortex, the areas that are used for the first time to perceive the visual information. We have these areas here at the bottom, and as you go up in the hierarchy, you start finding areas that have a more complex role in processing the information, in recognizing objects and images and all that. So this hierarchy goes in the sense of sensory areas projecting bottom-up stimuli to cognitive areas, and then cognitive areas can also send signals back via top-down modulation that could help us to understand, priors or attention and other phenomena. And this is all based on anatomical information, on structural information in the brain. Something very interesting that happened a few years ago is that people started to see some signatures of this directionality in the brain when you look at not at anatomical projections, but at functional features, at the basically electrophysiological recordings. And I'm just going to briefly mention two studies. One of them was by Van Kerkhove and colleagues, where they recorded the activity in V1 and V4, these primary visual areas, and they saw that when the information is going in the feed-forward direction, from V1 to V4, the electrical signals, the local field potentials, tend to show a very strong oscillation in the gamma range. Gamma range, for those who are not familiar, goes in the range of maybe 40 to 80 hertz, something like that. This is a very fast oscillation in the brain. On the other hand, if you look at the information that is transmitted in the opposite direction, from V4 to V1, that would be the feedback direction, there seems to be a very prominent role of what is called as alpha oscillations. And alpha oscillations tend to be around 10 hertz. Okay? The same thing was observed in another study considering more areas from Bastos et al. That I'm showing here from the laboratory of Pascal Fries, where they show exactly the same, that there's a peak if you measure it with this kind of tool that is the Granger causality analysis, pretty much a causal correlation between two areas. You see that there's a strong peak in the feed-forward direction in the gamma component, and if you look at the feedback direction, the peak is more strongly at the alpha or low beta component. So that's interesting because it means that in this kind of hierarchy, anatomical hierarchy that we have at the beginning, we also have some kind of dynamical signatures going on, and we can identify these gamma oscillations going in the bottom-up direction and these alpha oscillations going in the top-down direction. Since we have this information about the laminar structure from our anatomical data set, we can actually use a model to try to understand how these new interactions arise in cortex. So we build a model, let's call a multi-scale model that has 4 different levels of description. The first one is a classical excitatory inhibitory circuit, like the one that we were using before. Then we can use these to describe each one of the cortical layers that we are going to consider. We are going to consider supragranular layers and infragranular layers, and we can connect them using anatomical information. And then we can connect each one of the circuits in the areas, in the different cortical areas, according to the information that the anatomical data set is giving us. And then we finally put it together into our skeleton. So this is kind of a 4-level description that has computational predictions for each one of the 4 models. But for the sake of simplicity and to go to maybe the more interesting point, I'm going to focus only on the level of the interactions between B1 and B4, which are the results that I just showed you before. As I was saying, if you look at the results from these 2 studies, whenever there's information propagating in the feed-forward direction, you see a strong peak in the gamma component, in the gamma oscillations. And this is something that the model is also giving us. As you can see, this is the resting state, and this is the situation in which we have stimulation in V1. And as you can see, there's a strong increase, mainly in oscillations around 30, 40 hertz. This corresponds to the low gamma component. If you look at the other component, at the alpha component, we don't really have a lot here in agreement with the experimental data. We can also look at the other direction. If we remember, if we look at the feedback direction from B4 to B1, now we have a peak more in the alpha component of the oscillations. These are like 10 hertz. And as you can see here, there's a very strong peak with comparing rest and stimulation conditions for the feedback direction. So again, in agreement with the experimental results. We don't see anything, well, we see a small decrease in the gamma component, which is also something that they observe in the experiments. If you see the little blue shade over here, it means a decrease in the gamma component, but it's not important at this point. Finally, we can look at how the Granger causality looks like, and this Granger causality is basically the tool that they use to characterize this kind of frequency-specific interactions. And we can see that in both studies, they observe that the gamma component, in this case, the yellow curve, is stronger for the feed-forward direction. And in the region of alpha, Granger causality is stronger in the alpha region, so around 10 hertz. Same thing that was obtained in the other study, and same thing that our model is revealing. So it means that the model as we have it right now is able to reflect these functional interactions very well. But this is only for 2 populations, of course. What happens if we go to the full system? Because again, we have a large-scale brain network of the macaque that has 30 areas. So we can see what happens in more of these cases. And we, of course, see that now the situation is a little bit more noisy, but still the main pattern, the main idea is maintained. And as you can see in many of these figures, in the feed-forward component tends to have a peak, a very strong peak in the gamma range, and the feedback component, which is in orange, seems to have a stronger component in the alpha range. And this is something that, with some deviations, but it's something that is consistent across many of the area pairs that we have analyzed. And this, of course, gives us a new tool, a new trick to define a direction in the brain, a hierarchy, let's say. But instead of based on anatomical criteria, this hierarchy is based on functional criteria, on the activity of the different areas of the brain. And this is what people have called a functional hierarchy. So if we define a functional hierarchy in the sense that, let's say we have one area here at the bottom of the hierarchy, and then we have another area at the top of the hierarchy. In the functional hierarchy, this direction, the bottom up, would have a very strong gamma component, and in the feedback direction, it would have a very strong alpha component. Okay? If that is the case, then we can define an arrow, a direction between these 2 areas. And we can extend this to many areas and build some kind of ranking or some kind of hierarchy at the functional level. And this is something that Bastos et al. Were able to find experimentally, like we can see here. And we also were able to find it with the model. Again, the ordering of the areas is not exact, but the areas corresponding to different lobes are located in the corresponding branches of the hierarchy, just like they observe experimentally. And of course, we can also check if, going back to the first part of the talk, if increasing this kind of transient amplification due to the excitation inhibition balance, we have an increase in the propagation. And just like in the other model that we showed before, our laminar model is also showing some improvement in the propagation. Okay, so just to check that everything works as expected and our balance mechanism is also robust and holds in this case. Okay. So the final thing that I want to talk about is, and this is just one small part, because I talked about mainly dynamics of these large-scale networks. I told you how, according to these models, how the activity of different areas goes from one area to another, so how the signal propagation goes in these realistic networks. And we have discussed also what happens in terms of brain oscillations that are all observed pretty much everywhere in the brain that you put an electrode. You are always going to find oscillations of different frequencies, and we see that there's a little bit of an organization, hierarchical organization in this sense, and we see that our models are also able to reflect that. But this is just dynamics. At the very end, now at this final part of the talk, I'm going to tell you a little bit about how can we go from dynamics to function. And we are going to focus, we can focus on many different cognitive functions that we can model with these large-scale networks. But we are going to focus on working memory. What is working memory? Many of you will be familiar with this term, but those who are not. Working memory, if you allow me for the kind of oversimplification, could be understood as the RAM, the random access memory of the brain, some kind of volatile memory that we can use for a little bit of time before we forget. So it's not something like the memories that we remember from our childhood. This is a memory that comes and goes. And it's very useful because it's, but in particular, there are many types of short-term memory, but working memory is a very particular type of short-term memory in which we store this information in our brain for a few seconds, and then we manipulate this information to be able to do something later. Okay, so it's oriented towards an action at the end, a motor action. A classical example is this old studies when you basically show a monkey two possible spots. In one of them you have food, and in the other spot you don't have anything. You cover them, you cover the line of vision of the monkey, you wait for a few seconds, and then you release the screen, and then the monkey remembers where the food was, and is able to reach and grab the food. And this is a very simple example of working memory. You remember where the food was, and you're able to transform this memory into a motor action and reach out. Okay, of course, after 2 years, you won't remember this, but at the moment, this was very important to remember where the food was. And this particular cognitive task has been studied in many different ways. Classically, what people would do would be to insert electrodes in a region of the brain that is called the dorsolateral prefrontal cortex, here at the front of our brain, because this is where the first recordings showed that there's some kind of persistent activity going on during the delay period, during the period in which the monkey or the animal is basically waiting until he receives the go cue and he can reach the food or look at one particular direction or something like that. So for many years, people thought that working memory was mainly and uniquely happening in dorsolateral prefrontal cortex. Okay, and people attributed this effect to some very strong recurrent connections between neurons that you can find in dorsolateral prefrontal cortex. But with time, of course, people started to see that even though dorsolateral prefrontal cortex is very important for working memory, there are other areas, both in monkeys, in macaques, and also in humans. There are other areas in which you can also find activity that is related with working memory. And I'm showing you a figure of one recent review in which working memory associated with several things, including shapes, colors, sounds, numbers and different things are identified in different parts of the brain, okay, and also in humans. So it's not true that working memory occurs only in prefrontal cortex. It involves many different areas in the brain. But this, of course, this is of course a problem for the classical way that people thought about how working memory arises. So in the classical approach, people think that working memory can occur, this kind of persistent activity that the experimentalists observe during the recordings emerges because of local bistability, meaning that we have a dynamical system, which could be the neural network in dorsolateral prefrontal cortex, and for some regions of the strength of the synaptic connections, you can have not one, but several stable points, several solutions of the firing rate activity. In this case, for instance, typically this solution is unstable, meaning that it's not a solution that can be maintained over time. But you would typically find one stable solution corresponding to low firing rate activity and another solution corresponding to high firing rate activity. So it's this red line here and the red line here. As you're seeing here in the figure, this basically requires that your parameter G, that is basically the strength of local projection, has to be between these very narrow values. If you are here or if you are here, you won't be able to have this kind of multi-stability that allows you to store information in the brain. And this is kind of problematic because if we are going to assume that different areas in the brain are able to maintain information using working memory, we stumble about the problem that different brain areas are very different from each other. So some areas here would have strong projections, but some areas here would have very weak projections, very weak synapses. So it doesn't seem like all of the areas are going to be able to fulfill this requirement. So this is where large-scale networks come. And to solve this problem, we propose what is called the distributed working memory hypothesis, which is that instead of relying on local connections between neurons in dorsolateral prefrontal cortex, neurons across the brain form loops, large-scale connections, let's say big loops, between different areas, meaning that they are not going to be helping each other to maintain information only locally, but also globally. If we do this, the space in which we are able to obtain this kind of bistability goes from just a little range here to a broad range in our parameter space of the system. Meaning that now areas that were originally too weak to contribute to working memory, now they are able to sustain information and contribute to working memory. And we can even consider the extreme case in which we can assume that there's not a single area in the brain which is able to maintain information using persistent activity by itself. On the contrary, all areas in the brain would need the help of other brain regions to sustain this information. And this is an extreme case. But even in this extreme case, our model seems to be able to work, as I'll show you in a second. A couple of details about the model. As I said, the main ingredient is this kind of big loops that we are going to assume. And we are going to constrain these loops using the anatomical data set from the macaque that we have and that we are already very familiar with. So now we can model each one of the areas as a small neural circuit in which two excitatory populations are competing with each other. Let's say one population wants to remember a stimulus, let's say a color, blue, and another one wants to remember the color green, for example. So they are in direct competition, and they are going to allow us to see how the model is able to maintain information. And of course, we also consider that areas in the brain are different from each other. And we introduce this in the model by including data on the density of dendritic spines, basically on the number of synapses that a typical neuron is receiving in each one of the areas. And this reveals some kind of gradient from a collection of anatomical studies. So we can introduce that in the model as well. And we have our results, very simple results of the model. In this case, for example, we are modeling again the cortex of the macaque. So we give our virtual macaque a visual stimulus, which is going to trigger a strong activation of the occipital cortex of the primary visual cortex here. And then this activity propagates to other areas. And during the delay period, the phase in which the monkey has to wait, the information is maintained in this set of areas across the cortex. And this is the extreme case. This is the case in which if we isolate these areas from each other, none of these areas is able to sustain information by itself. It's an extreme case, okay? But if they work together with each other, then we are able to maintain the information of the visual stimulus. And you can see here some traces of the firing rate for neurons in different populations. And we can see how the activity is maintained via this persistent firing in each one of the areas. And this represents the memory that is transiently stored in the brain. I can tell you a little bit more details, but something in particular is that this seems to reflect some kind of change, some kind of bifurcation in the cortical space, in the sense that you either have areas that do not participate in the working memory, or you have another phase in which areas participate in maintaining this information. And of course, some areas participate with a higher firing rate, some areas participate with a lower firing rate, but there seems to be a jump here, a change in the behavior of the system, a bifurcation. And you can move some of the parameters of the model just to see that this bifurcation is going to be there no matter how much you move the parameters of your model up to a reasonable point. And then I think I'm closing to my finish, so I'm just going to tell you what happens. Of course, if we have this model for working memory, we can use it to study many things, but I'm just going to show you the typical example. If you have a model like this, you basically want to play with him and try to lesion some of the cortical areas to see what happens, right? So what happens if I shut down one of the areas? And this could be reflecting pharmacological procedures to block the activity of the neurons in one particular area, but not in other areas, right? Well, what we see is that, for instance, if we had the scenario before with visual stimulation that goes to later to this distributed activity during the delay period, we can, of course, try to do the same, but first we can lesion one of the areas and see what happens. And we are going to lesion one area, 24C, that is also known as anterior cingulate cortex. It's an area here in the frontal region, frontal limbic region. And we see how lesioning one area, in this case, blue is the control situation, and red is the case of the lesion. Of course, this area stops working in the lesion because we are basically blocking all the activity here. But the activity in the other areas, it doesn't seem to be affected a lot. Meaning that in this model of distributed working memory, memories are able to be stored in the neural network in a very robust way. So if we lesion part of the brain, we are not going to lose the information. Of course, this happens with some areas, with one area, but there are some areas in which lesions actually shut down the activity in many or even all of the cortical areas considered. And if you remember, I was telling you that dorsolateral prefrontal cortex has classically been seen as one of the most important areas, one of the most important cortical areas for working memory. And our model reveals that for a reasonably ranged set of parameters, lesioning this dorsolateral prefrontal cortex area shuts down the activity. And as you can see here now, none of the areas is able to maintain the information for so long. All of them decay after some transient. And there's a lot more things to say about these things, but looking at the time, I think it's my time to finish. So I'm going to go over the conclusions very quickly. So what we have done, what I've shown you in this presentation are basically a new set of large-scale brain models in which we use simplified assumptions for the dynamics of the different brain areas, like firing rate models, even though at some point we have also considered more detailed things like spiking neural networks. But importantly, instead of just putting them together and assigning random values to the connections, we have constrained our model with real anatomical data obtained from neuroanatomy experiments. And if we do that, we are able to answer a lot of questions that we had, and some of them that we didn't know we had. Like, for example, at the beginning, we were very happy with our models for how activity propagates across several areas of the cortex, and then we found out that when we put these models into a realistic brain connectivity, they don't work at all, and we need to consider extra ingredients in our model, like the transient amplification given by excitation-inhibition balance. We have also introduced details about laminar structures in cortical areas, and we have seen how this is able to reproduce some of the findings regarding oscillations and regarding how gamma and alpha oscillations are able to show some kind of new way of ranking the different areas, a new cortical hierarchy at the functional level. Okay, and then we finally went a little bit beyond dynamics, and we started to consider function. We started to consider a particular cognitive function known as working memory, and we see how having a large-scale network, anatomical data to constrain this network, is able to tell us new mechanisms that rely on something beyond the well-known classical local rules of working memory and into the new realm of distributed computation and large-scale neuroscience. I'm just going to finish that. If you like this kind of research, now at the University of Amsterdam are developing similar lines, but focusing on mice. Looking at models of a mouse brain is interesting and convenient from many points of view. For example, you have a lot of anatomical data, much more than for the macaque, that you can use to constrain your models. So in principle, the models of the mouse will be more detailed and more accurate than those of the macaque that I just showed you. And you have people now doing high-density recordings with neuropixel probes and all kind of very fancy recording techniques that people can use in mice. Also, the classical optogenetic recordings and stimulation can be used here. So it's a very versatile system. And also, mice are very easy to train compared to, for instance, with macaques. It takes a lot of time, a lot of years to train a monkey to do a task so that you can know how the brain performs a particular computation. But with mice, it's a little bit more simple. And I'm just going to put you some references in case you are interested. These all published in a few journals here. And these are the people who contribute to this work. Joe Murray, a professor at Yale University, Madhura Joglekar. I think I have to update this because she's not at Koran anymore. She's working in a company. Robert Young, working at Columbia University. Henry Kennedy, providing the anatomical data set from INSERM and University of Lyon. And Xiaoying Wang at New York University. I think that's it. I'm ready for any kind of questions and discussions. Looking forward to it. I think I'm gonna.
Thank you so much. Thank you so much, Prof. Mejias. This was really fascinating.
Thank you.
So we have about half an hour left for questions. So I think what I'll do is, if you don't need your slides to answer them, maybe I can just minimize the screen for us. There we go. So I think we'll start with the first three and see how far we go. I think we might only get to them. So the first question we have is from Louis, and he is asking you, how does one find these hierarchical graph structures? Do you stimulate some areas and find other areas that spike, or do you analyze the structure of the physical brain?
Well, that's a very good question, because there are two types of structures, let's say hierarchies, that I mentioned here. I'm assuming that you are referring to hierarchies. If you are referring to the structure, to the connectome, you basically do anatomical recordings. You basically inject a tracer, that could be a virus, in one particular part of the brain, and then you track how this tracer, this virus, goes to other areas of the brain. And then you can see this once you cut the brain a little bit. So that's how, and then basically once you cut the brain in slices, you do the histology, you can count how many neurons in this area were projecting to this area. Okay, that's one of the ways people in neuroanatomy build this kind of large-scale networks, the structural part. Okay, but just in case you're referring to the other things, the structure about the hierarchy is, if you're referring to the one for the anatomy, is basically using similar approaches. If you're referring to the functional hierarchy, this is basically this is basically found out measuring the activity of cells in different areas, right? And then you see, you basically record the activity of this area, and then you see that this one, whenever you consider the information transmitted from this area to this one, you have oscillations in the gamma range, and in the other direction, the oscillation seems to be more in the alpha range. So there's basically two structures here. One is the anatomical structure that you can determine using this kind of injections, these tracer injections, and the other one is the more novel functional hierarchy that you find by recording the activity of neurons in different brain areas and doing a little bit of data analysis.
Okay, perfect. The second question we have here is from Amit, and he wants to know, how do you extract activation thresholds from simulation? Are they predetermined or somehow emerge as a result of the simulations?
Sorry, can you repeat? I kind of broke down a little bit the first part of the question.
Yes, no problem. So he is asking, how do you extract activation thresholds from the simulation? Are they predetermined or somehow emerge as a result of the simulations?
They emerge. The activation thresholds are a very simple concept. You can of course define it in many different ways, but in this model, which is kind of simple, we just assume that each one of the areas has some kind of range, has some kind of dynamical range. They can be non-firing or firing very little, and they have also a maximum firing rate. So we can see, for example, how much input do we need to put this area into a 50% activation. Okay, so basically they're halfway towards the full activation. You can define activation levels like that, and then you see that the amount of input, the amount of visual input that you need for early sensory areas to reach this point is very little compared to the amount of input that you need for areas in prefrontal cortex, for example. And again, this can be defined in many different ways. I think the activation thresholds that they use in the experimental study that I showed here define the activation threshold in a slightly different way, but they are consistent. We later try the definition that they have, and we basically see the same. So it's something very flexible in this sense.
Thank you, Prof. Mejias. And thank you, Amit and Louis, for your questions also. I'm now going to invite Dimitri on screen to ask his questions to you himself. So I'm sending him an invitation now. Dimitri, you should be receiving an invitation shortly. Okay. Okay, he says so far he has not. Okay, I'm going to try again. Oh, you're still not receiving it? Okay, he's saying he's not receiving it. So I'll just continue to ask this question then also. So Dimitri wants to know the result about the interaction of two EI circuits and the relation to oscillation frequencies. Are there analytical results in this direction?
Not sure if I got the final part. Would analytically solve, you mean solving the you mean doing an analytical derivation of the oscillations instead of simulations?
Yes, he says sort of.
Okay. So maybe I can try to give you an answer, and then you can tell me if that's what you meant or not. Maybe we can do the discussion this way. So typically, and this is something that is known for models of small populations of neural networks. You can have spiking neural networks or firing rate. Typically, when you put a population of neurons, excited or inhibitory neurons, there are several mechanisms that can make these oscillations arise. Okay, and they go with, for instance, ping mechanism, which is the one describing oscillations that arise because of interactions between excitation and inhibition. And there's also the ing mechanism. That is basically oscillations that arise just because of interactions between different inhibitory neurons, and then excitatory neurons are basically following up. Okay, so the properties of these oscillations are going to depend on the parameters of the system. Okay, if, for instance, some parameters that are very important for these oscillations are the strength of the connections between different populations. Okay, if you consider some level of, for instance, if you consider that the projections between excitation to inhibition and inhibition to excitation are very strong, you are very likely to have this kind of oscillations. And if you have other kind of configurations, maybe you don't have oscillations, you have another type of dynamics. So this can be studied using simulations, but this can also be derived analytically. So just doing a mathematical derivation, pen and paper, and you can have the properties of your oscillations, including the frequency, the amplitude of these oscillations. So this is something that is very well studied at the level of the classical neuroscience models. Now, that's the reason why we use these kind of things to build a large-scale model, because we wanted to build on something that is very well known and very well established, just to make sure that we have our feet on solid ground, basically. Okay. I don't know if that's more or less the answer that you were expecting. Can you tell me yes, no?
It seems like Dimitri is saying it is well studied for single E and single I populations, but not for several.
Single E and single I population, but not for several. I guess it depends on the level of the system that we're considering. But if you assume this kind of firing rate models that I was showing, it's also well described for E and I populations. And for that, you can check, for example, the Dayan and Abbott book. They have a very classical example with numbers and all that, when they actually show oscillations using these models. It might be different, of course, if you're considering models that are very realistic. You have two or more interconnected EI populations. That's true. Yeah. Okay, now I get your question. Yes, if you have two or more interconnected EI populations, then analytically it's not as clear. It's clear if you consider, for example, that the connections between them are weak. And then you have all this theory about the weakly coupled oscillators from the classical papers of Kuramoto and these things, where you can actually show analytically that there's going to be some kind of global oscillation arising. But that's, of course, in simplified models and assuming that the connections are very weak. If the connections are stronger, I think you can use some perturbative analysis, but the extent to what this gives you a good, solid analytical result, I don't remember at this moment. But at least there's some indications in this sense. And again, once you go to something complicated like these networks that we are considering that are constrained with data and all that, then the analytically approach is very difficult to get. We are exclusively relying on simulations in this sense.
Thank you.
No, so yeah, to answer to these things, no, all the work that we did was in simulations in this sense, so we don't have analytical work looking at these things.
Okay. I apologize for having to do this discussion this way. Sorry, Dimitri, and sorry, Professor.
No problem. As long as we are able to interact, that's okay.
Yes, that's true. Thank you for your question, Dimitri. We are now moving on to the next question. We still have some time left. This one is again from Amit, and he is asking, in trying to understand the simulation a little deeper, what are the differences in the way you model each neural network of a given area, example V1, V2, etc.? What are those models based on? Beyond the number of connected synapses, are there other differences between areas? And finally, are there real features that are difficult to model and hence are left out?
Oh, these are a lot of questions, so I'm going to try to answer all of them, and if I forget one of them, please tell me. So mainly differences in between different cortical areas. That's a good question. It depends on the part that you refer to. So basically, I was explaining three different things. For example, for the model of cortical layers, this is very. This was the first model that we developed, even though I talk about it in second place. So in this model, we have very little differences between different cortical areas. We have, of course, differences in the way they are connected to each other. So for instance, you're going to have one particular area that is receiving projections from other, target one particular layer and have one particular level of scientific strength. And this is going to be different than from the other one and from the other. But if you look at the individual areas, for this model, they are exactly the same. Okay. Now, if we go to the two other models that I presented, the one for signal propagation and the one for working memory, then yes, in those models we have cortical areas that are modeled in different ways. And in particular, the equations are the same, but then of course the parameters that we use are slightly different. And they basically go in the same direction. The idea is that both in the signal propagation and in the working memory paper, we use the information about the dendritic spine densities that I showed at some point in the lecture. And if you remember, this density of spine counts is very low for early sensory areas like B1, and is very high for other areas like prefrontal cortex. So these numbers can be implemented in the different firing rate models that I'm using for the different areas, right? For example, if we are assuming that there's a lot of synapses, you can assume that the overall synaptic strength is going to be larger, if only just by having more synapses, right? And this is a naive assumption, because then, of course, this could, in principle, compensate with, for example, an area that has more synapses, but the synapses themselves are weaker. Okay, we don't know about that. We have some reasons to think that this is not the case, because then they would compensate and they would account to the same level of average recurrent activity. And there are some evidence looking at the time scales of how different activities of different areas behave. And these kind of differences in time scales can be explained by this kind of, differences, area-dependent differences in the level of recurrent connectivity. So it's not a proof that is solid still, but there's some evidence pointing towards the direction that these kind of differences in the level of dendritic spikes reflect, up to a point, differences in the strength of local connections. And this is the main factor that we have used to establish differences between many cortical areas. I'm not sure if that answered the whole question, or I'm still missing something.
I think we can leave it at that. That was a very, very long question. Thank you so much for your question, Umit. You can just pop us a comment in the comment section if you want to know anything more on this particular question. Then we can move on to the next question. I'm going to invite Siobhan to ask this one.
Okay. So the question is, I didn't quite understand the argument used to argue that classical bistability is bad. The region of bistability can be pretty large if G value changes only very slightly physiologically. Would you mind commenting on that, please?
Yes, that's also an interesting point. One of the, yes, I mean, this point is kind of long to argue, so that's why I basically say that this is a very small range. But of course, this is a parameter. We can basically stretch the range a lot. There are also some considerations to think that this kind of model, bistable model for working memory, might not be adequate for all the areas in the brain, and that's pretty much the point, right? If we assume that this area, this bistability mechanism, could explain the persistent activity related with working memory in all brain areas, then they should behave more or less the same. But it doesn't seem to be the case. Like, for example, there's evidence that there are areas in which persistent activity can be turned down by using distractors, for example, visual cues that are trying to distract the monkey or the human from the cue that he has to attend to. And these kind of distractors are kind of difficult. These kind of differences in how distractors affect, for instance, the activity in prefrontal cortex or some other areas like parietal cortex are difficult to justify if we assume that all of the areas behave via this kind of bistability phenomenon. And that's something that if I think we have one of the figures of the manuscript is addressing exactly that. So if you're interested, just go into the paper, and there's one of the figures in which we show how having a distributed network for working memory can explain things about distractors that cannot be explained with classical models of working memory. But that's, for example, another consideration.
Awesome, thank you.
Welcome.
Thank you so much. It seems like we have Dimitri online. Welcome, Dimitri. So now you can ask your next question yourself.
Yeah. So basically, it's a little bit elaboration of what you already answered in one of the first questions. So is there other studies directly comparing something that you get from range of causality and something that you get anatomically? Of like when they confirm each other and when they contradict each other?
I'm not sure if I got your question. You mean comparing the Granger causality with the anatomical hierarchy?
So I mean, you have the functional connectivity, like from Granger causality or whatever else you meant by that, and you have anatomical connectivity, and you can try to compare, well, to some extent between them.
Yes. Yes, they are not the same, right? I mean, they are two different things. That's the first thing that we can say. They are not the same. They are called hierarchies, but they are different things. One of them is based on anatomy, and the other one based on function, on dynamics. They are not the same, and they don't have to be the same in terms of, for instance, areas that could be higher in the anatomical hierarchy could be very low in the functional hierarchy.
But
right?
Do you have like good, really striking examples of something that is really strongly connected, let's say, anatomically, and not connected at all functionally, or vice versa?
Yes, there are examples. I would have to go into details, but I think one of the examples is frontal eye fields. Frontal eye fields is one area that has been classically assigned to the higher areas in the anatomical hierarchy, but then in the functional hierarchies, it has a role that is more on the low level. And there's a lot of potential reasons for that. I don't have a full explanation, but that's one example that we could think of a strong disagreement between the functional hierarchy and the anatomical hierarchy. And it's also interesting, let me say, because what they observe also in the functional hierarchy is that, of course, it depends on the dynamics, so it can change. In some situations, you would have areas a little bit higher in the functional hierarchy, and in some situations, they would be a little bit lower. And in the Bastos paper, for instance, it is shown how they, depending on the data that you use, if you consider the data before the monkey observes the visual cue or after it observes the visual cue, the position of the areas in the hierarchies, in the functional hierarchies, changes a lot. Well, a little bit. Okay, and one of the areas that changes the most are actually frontal eye field areas. So I don't have a definitive solution for that, but frontal eye field seems to be one particular example of this kind of discrepancy between the two.
Awesome, thank you.
You're welcome.
Thank you, and thank you, Dimitri. So we have our final question left, and we have Edward on screen to ask it here. Welcome, Edward.
So this is probably a very naive question, but with these spiky activation models and average activation sort of networks that you have, would they be able to support some sort of optimization or training mechanism that we know for sort of conventional networks, something like the back propagation of the brain or something?
Okay, good and difficult question. Let's say if we have to, I think can you? I kind of hear myself. Can you shut down the microphone? Yes. Okay. Perfect.
Yes.
So this is tricky because these kind of models, if you look at the equations for these models, they are very, very simple. Okay, there's no any kind of training process. There's anything particularly difficult. They are basically differential equations, dynamical systems that behave the way I showed for a particularly large range of parameters. So there's no fitting, there's anything like that. So one has to think about these models as some kind of first impression, okay, some kind of broad description of what's happening. Now, the idea, in order to, for instance, if one wants to make this compatible with models in which you can use back propagation to do a finer training of the model and make the model able to, for instance, to categorize particular inputs and all that, then a good approach would be to take this model as a skeleton, as some kind of a broad description, and then add these details on top of that. This, of course, I'm saying it like it's easy to do, but I think it's super difficult to do. I don't think that this kind of, there's very few people that have been able to make solid connections between these dynamical system models and the ones that you more focus on training. There are a lot of studies on that, so it's kind of an emerging field. But they are not the same, and they are difficult to merge. So that's something I would say that, I mean, the models that you can train usually are more convenient if you want the model to do something, if you want to network to do a particular task. The models on dynamical systems reflect a little bit better what's happening at the level of individual neurons and the physiological properties and all that. So each one has their pros and cons. Now, if at some point we're able to get a hybrid or something that is able to, then it would be awesome. And I know that there's a lot of people working these kind of things. So I hope that in the next years we start seeing results in this sense.
If I can maybe ask one more additional question. I think you answered it before already. I just want to make sure that I understood it correctly. But for the models that you have now, you sort of approximated the connection strength. I know you said something about traces, but did you look at the physiology, like I don't know if there's a thickness associated with the neuron or something like that, but did you get the connection strength from the physical properties, or how did you how did you quantify that when you started?
Yes. The way to obtain. Sorry. Can you mute the? Okay, perfect. So the way to obtain this is very simple. What they are, again, I'm not neuroanatomist, so I'm just telling you a very crude description. But there's different ways to do it. But in short, the way that the method that these people use is to inject a tracer, a substance. Sometimes it's a virus, sometimes it's another thing that can travel from one area of the brain to another, right? Because let's say you have one area of the brain that is connected to another one, and you have neurons that have synapses that project into this area, okay? So then if you inject this tracer and the monkey, and then you sacrifice the monkey and cut the brain in pieces, you will see the tracer in this area of the brain, but you injected it on here. So it means that it has been traveling here, okay? So then you can, since this is basically at the level of individual neurons, you can count the number of neurons, okay? And that's how we give some kind of a proxy of the synaptic strength. We don't really know the synaptic strength. We know the number of neurons that are projecting from one area to another area. And then, again, we assume that this has a correlation with the real strength. It doesn't have to be this way, but this is the best information that we have at this moment. And that's what we can use for the models. So then another extra thing would be to check the real strength of the synaptic projections between these two areas. But for that, you need to do a systematic electrophysiological study across all these brain areas. It would be fantastic to have it, but up to my knowledge, I don't know any of these studies. I mean, I know for some areas, but I don't know any study that cover 30 or more cortical areas of the macaque, like the database that we have right now. Yes, perfect.
Maybe while my microphone is on again, it kind of blows my mind. So these tracers physically move through the neurons and then go to the next one, and that's how they end up with the other region. Maybe you could give an estimation of like what the propagation time is that you give them. Is it sort of, it's seconds or hours, or how long does it take one to get from here to somewhere else? Or does it happen with act?
I don't know, actually. I don't know, because this is a very artificial process, and I mean, the moment is enormously invasive, right? So the moment you do the tracer injection, then you have to sacrifice the monkey. It's something that you cannot, make compatible with other recordings or something like that. So I don't really know the process, and I don't really know the time scale for that. But I think it's something that should be. Maybe even someone in the comments know. Some of the people they can comment, because this is the kind of thing that a neuroanatomist should be able to tell you a little bit more. But I really don't know the answer to that. Maybe it's just a matter of digging into the papers.
Cool. Happy.
Okay. Thank you. We have some answers here. I was waiting for these guys. Jota also works on these things, and Xiang as well. And they are replying. They are also in the lab of Xiaoying Wang at NYU, so they know pretty much all of this stuff. And they say that it could take a few weeks. So it's a very slow process. If that's useful.
Okay.
Thank you to
very
everyone in the
much.
comments also for covering that.
My pleasure.
Okay, before we kick off the thank you, I'd just like to draw your attention to some events that are happening. Neuromatch has a summer school and conference coming up, and we definitely recommend that you check that out. And then we'd just like to give Prof. Mejias a virtual round of applause for a very good talk on this Wednesday evening. Thank you for attending. We hope to see you at future events. So please remember to follow our page on Crowdcast so that you are able to see future events. And then last but not least, we would like to thank our sponsors at Stellenbosch University and the Biomedical Engineering Research Group. And we look forward to seeing you on 6 May for our next event. Thank you and good night.
Bye-bye.